Hey everybody. So in this video we're going to learn about how DNA is actually packaged and kept in the nucleus of the cell. So we know that DNA is found in the nucleus, and we also know that the DNA is kept as separate molecules, and they're called chromosomes. And we've learnt that humans have 46 separate chromosomes in their cells. But cells of living things actually organize those chromosomes and keep them packaged in a certain way so that they don't all get knotted together and unusable. Think like a bowl of spaghetti. So if the chromosomes weren't packaged together, they'd become tied up, they wouldn't be able to be accessed by proteins, and it would be really hard for the cell to control which genes it wanted to turn on and off. So cells need to organize and package their DNA. For those reasons. And what scientists have actually figured out is that each cell has about 2 meters worth of DNA. And if you think about a cell, it's only around a few micrometers long. Somehow it has to fit 2 meters worth of DNA just inside the nucleus. So a few different mechanisms have evolved to be able to keep that DNA organized. And here's a diagram showing the basic way that cells do it. So we're starting over on the right here. Here is the DNA double helix. And if we zoom out a little bit more, you can see that that DNA, it actually starts to wrap around these proteins here. And we'll learn more about them later. But they're called histone proteins. And you could think of them like little beads, and the DNA is like a string. So the DNA starts to wrap around these histone beads, and it makes these larger beads here. And then those beads in a string, they start to coil around each other to make these coils. So think of like an old telephone wire, how it's wrapped round and round and round in a coil. And then if we zoom out again to the next level back, those coils, they actually start to coil around themselves too. And when there's 2 levels of coils, that's called a supercoil. So that's the 4th level of folding. And then if we keep zooming out again, those supercoils, they fold up into these classical X shape that we know and we recognize as chromosomes. Remember, chromosomes don't always form that X shape. It's only at a certain time in the cell cycle. So you can think that DNA will either exist in stage 4 or stage 5 here. Exists in either of these forms. Normally it's stage 5 when we can collect it and take photos of it in the microscope, but usually it's in the stage 4. It's been coiled up twice as well. So when we first learned about chromosomes, we said that they're just a single DNA molecule. Well, that's kind of right, but also we need to modify that now because we know that chromosomes are actually made of these histone proteins too. So the proper name for the material that makes up chromosomes is called chromatin, with an N. So chromatin, it's the name for the combined DNA and histone protein all together. So that's a better definition for chromosomes. They're made of chromatin, which is DNA and histone proteins all wrapped up together. And so in this way, cells can keep their chromosomes neatly packed and they can access particular genes when they need them.